Hello and welcome to the University of Calgary webinar, Cracking the Genetic Code for Better Health. My name is Jennifer Alford and I'll be your moderator today. We're going to meet our scholar in just a minute, but first a little housekeeping. I want to give you a little information about what we're doing today and how the whole thing works. The webinars that we are having here today, they feature a series of University of Calgary scholars, and they bring us a summary of their research. We invite you to ask questions throughout the webinar. Just type it in that little screen to the left, and we hope to get to as many of your questions as we possibly can throughout the next hour. We're already getting questions, so I suspect we're going to have quite a few. And please, if you're on Twitter, keep the conversation going with the hashtag ExploreUCalgary. Now, we're going to meet our scholar. Dr. Francois Bernier is an expert in genomics and the head and associate professor in the Department of Medical Genetics here at the Cummings School of Medicine at the University of Calgary. Welcome.
Hi, Jennifer. It's good to be here.
Well, thanks for being here with us today. We are having a little bit of trouble finding our slides here. There we go. Okay, I think I figured it out. Here we go. We've got quite a lot of ground that we're going to cover today.
Yep, for sure. So we're going to start by talking a little bit about the human genome and how we sequence the human genome, the impact of genes in our health, and sort of the types of things you might learn about ourselves if we were to sequence our genome. And then end by talking a little bit about the concept of precision health. You know, where are we heading in medicine and what's the next era going to look like? So I'm going to dive right in and continue on. And to start, we're going to ask our audience to help us out here a little bit. So this is a poll for you. And the first question is, and you'll get a chance to answer it in a second, is genomic sequencing should be standard medical procedure once it becomes cost effective. If someone is predisposed to health risk, they should know about it, and so should their doctor. So I'm going to open up the poll, and you can see on your screen now that you can pick your answer and just hit submit, and then I'll show you what the results look like.
A pretty big question.
It is a big one to lead off with. It'd be interesting to repeat it at the end and see if it changes.
That's a great idea, yeah.
We'll see if we get time.
There's so many different factors that come into play as you think about that question. All right. I want to know what you'd say there, too.
Okay, so there are your results. So about 80% agree, about 20% disagree. This poll was originally run as part of a Globe and Mail series on genomics and health, and you can see the Globe and Mail results here, about 76%. So pretty similar. And what that tells us is that there is an interest out there in understanding what the role of our genome is in our health. And so let's continue on. So I often get the question of, what is our genome? And I describe it as the series of instructions that our body uses to go from a single cell to a baby. So we need to use those instructions to develop. We then use those instructions to grow into an adult, but we also use them to renew ourselves. So minute by minute, second by second, all of us are sitting here using the information from our genome to maintain glucose levels, to create memories, to run the electrical system of our heart, to renew cells that are dying. So it's an incredibly dynamic system. So if you think of it, we're completely self-contained. All of the instructions we need for life and to sustain life is encoded in our genome. It's an incredible structure as well. So if you think of in each one of our cells, we have 23 chromosomes, these sort of bundles of genetic information. But if you pull on those, you're unraveling the DNA. So we have 3 billion letters of genetic code in each one of our cells. And those encode for about 20,000 different genes. We have about 4,500 different genetic disorders that result when you have changes in those. So the genome itself is an incredibly complex structure.
Mind-boggling, really.
Yeah, it is really quite remarkable. The amount of information we encode in it and how we interpret it is still being sorted out in many ways. So the largest scientific project ever was the Human Genome Project. It was a multi-billion-dollar effort to sequence the genome once. And 10 years post that success, in 2001, Nature published this article saying the future is bright. It was bright for a number of reasons. It was starting to be that, we were understanding the complexity of the genome and how it applied to health and understanding biology, but also because a transformation had occurred. And the transformation was that these instruments called next-generation sequencers were now coming online. We have some of these at our Cummings School of Medicine Center for Health Genomics and Informatics. They're kind of the most complex bread maker looking instruments you could ever buy. But these are massively parallel sequencers. So in the past, what you had to do was sequence small fragments of DNA, basically one at a time. So to do a genome, you had a multi-billion-dollar, 20-year project. These machines sequence millions of fragments in parallel at the same time. So they're revolutionary in their ability to generate really whole genome sequencing. You can sequence the genome of any organism now for a relatively cost-effective process. And there's another generation coming, and I love looking at these things. So we talk about nanotechnology sometimes, microscale, nanoscale instruments. And this is one that's developed that is a nanoscale sequencer. We'll call it the third generation or next generation for the Star Trek fans out there. But these things are tiny pores in the membrane, and they let the DNA sequence rip through at an incredible pace. And as each one of those little bases is going through, you can read them. And if you look at the little right-hand, or I guess left-hand bottom corner there, there's a little port on that instrument. That's a USB port. So you actually plug these into a laptop. I think the next phase of technology change is going to mean that these things are going to be available in offices, and small labs, and, attached to iPhones. This instrument went up on the space station. So although the technology was truly transformational, it's really the cost change that made the biggest difference. So if you look at, 10 years ago, this was still a $10 to $20 million project to sequence a single individual's genome. And now you do that for less than a few $1,000s. The cost predictions are, the $100 genome is going to be around the corner. And not 10, 20 years, 5, 10, maybe less. So this has really brought genomics, the concept that we can read this information, into the limelight. For me as a clinician, it allows us to think about diagnosing patients that were undiagnosable before, to treat patients that were untreatable, and to solve mysteries of things that were really impossible to solve before. So where I'm going to head now is we're going to talk a little bit about the impact of genes in health. And this is kind of my genetics 101 slide for people who kind of cringed around their biology days. So I think of pretty much every health condition can fit somewhere in this triangle. There are things that are caused at the top of the triangle by having one gene that's abnormal. There are things at the bottom that are kind of a combination of many genes. You could use height as an example. It's not just about one gene that creates our height. It's many genes. And then there are obviously things that are non-genetic. And then lots of things interact, right? The combination of gene and environment interaction. So in a somewhat simplistic way, then we can think of single gene disorders as these rare disorders that affect, up to 10% of the population. So they're individually rare, but there's a lot of them. And things like cystic fibrosis, sickle cell disease, the muscular dystrophies, breast cancer risk, et cetera, can all be caused by a single gene. And about half of us will have some form of health issue at some point in our life that is, where genes played a contributing factor, interacting with other things. So these are the complex traits. And we know those running families, the family that has, too much hypertension, too much heart disease, too much cancer. It just seems to be present in that family as a tendency. So most of what I'll talk about next is giving you examples of how genomics is giving us insight into these disorders, and particularly using kind of these rare genetic disorders as an example. So these rare diseases, I say they affect a large number of people. We define them as less than 1 in 2,000 individually. Many of them, most of them are truly genetic. Some of them are not. Many of them affect kids. I'd say these are some of our most chronic, difficult to deal with disorders in many ways because we don't have treatments for many of them. And we know the genetic cause for about half. So there's still a significant effort to find a diagnosis in many of our patients. And it's remarkable to have to sit down with the family and actually tell them, I don't know what your child has. We don't actually have an answer for that. Well, for an adult who's been struggling their whole life with a chronic disease and don't actually have an answer. So genomics gives us an opportunity to try and diagnose these, and I'll talk about one of the first patients, or actually the first patient who had their whole genome sequenced, all 3 billion letters at the Cummings School of Medicine. And this was a young man that I saw in my clinic who had recurrent infections. And what I'm showing you on the graph here is white blood cells that we use to fight bacterial infections were all low. They're all below the line there. And they were going up and down a little bit. He also had some other medical problems that were making us wonder whether he actually had a genetic cause for his recurrent infections. And his sister had the same thing. So a pretty good clue, 2 family members with the exact same pattern. This is maybe one of the most remarkable things I get to do is actually just stare at genome data. And we don't look at it in this format very often, because this is the first about 2,000 letters of his actual genome data. So it's this repetitive patterns, feels somewhat random of ACGT, the chemical codes of the DNA. But there's so much of it. If we were to sit here and kind of have him flip through the rest of his genome, we would be here for the rest of our lives, trying to get through 3 billion letters of genetic code. So obviously we have computer programs that dive into this data, start analyzing, and start saying what are segments of genes, how do those segments compare to a typical person, and are there any differences? And for him, if you hit a particular chromosome and go down about 17, 18 million letters down, there it is, that little letter A. There should be a G in that location. And that's a mutation. This region of the genome actually codes for a really important protein that controls the development of white blood cells. So he has an underlying genetic disorder in his immune system, and it's creating a risk of having low white blood cells, like bleeding disorder, and his sister actually created a risk of inflammatory bowel disease. So he's got an immune dysfunction. And, what's striking about it is, again, it's that one letter change that can have such an impact because it destroys the code that body needs to create that critical enzyme. So he's actually doing fine. He's on a medication now that's boosting up his white blood cells, and he's planning on starting a family of his own, and his kids will not be at risk of this. And so we've been able to give him an answer and try and help him understand how to get better treatment, and also how to understand what the risk of his children having the same condition.
And is that something that you couldn't have done without this advance in technology?
It would have been difficult for him. I think, in fact, he'd had the disorder. We knew he had recurrent infections for most of his life, and it wasn't until the age of 20 that we were able to throw an answer at the story. So it would have been difficult. And we'll see other examples of sort of how difficult it can be to arrive at answers for individuals. So the next young man I want to talk about is Madden. So we first met Madden in the healthcare system when he was quite young, actually, just after birth, a few months of age. He started developing really severe eczema. And a lot of kids have eczema, the itchy arms and kind of behind the knees type of stuff, and it goes away with creams. But, his eczema was really quite striking. It was starting to be head-to-toe eczema. It was difficult to treat. And eczema can be a sign that the immune system isn't working normally. That's why a typical child might get eczema. But his eczema was too extreme for that. And by a year of age, he had enough other issues going on that we were starting to wonder, is there something also wrong with his immune system? So by 2 years of age, he became actually quite ill on us. So now he was very small for his age. And he was small because his own bowel, the sort of part of the body that's supposed to be absorbing nutrients, had been attacked by his own immune system. So he had an enteropathy. He had the inability to absorb nutrients. And he developed a really serious infection. So he was admitted to the Children's Hospital, and we were able to kind of clean him up a little bit in the sense that, we could get the infection under control, and we were starting to give him better nutrition. But the real problem we had is that we didn't know how to ultimately treat him. And for kids who have these immune disorders, sometimes the ultimate treatment is give them a bone marrow transplant, which means you're going to remove his own bone marrow and then get a matched donor and give him an immune system. So it's a dramatic treatment, but we weren't really sure what his diagnosis was. Was he going to be okay without it, or did he do the bone marrow transplant? So he'd been admitted for many months. He had probably the best we could throw at him, including a large amount of genetic testing, and immune system testing, and GI system testing, and test after test. And the families describe these as diagnostic odysseys, that time that people spend, trying to find an answer for their child. And we remained without one. So we enrolled him in one of our research studies in the Alberta Children's Hospital Research Institute that had him go on to one of the sequencers there, and his DNA went on a Monday. By Wednesday evening, we had sequenced all 4,800 known genes that cause any disorder, so we just basically said, let's do everything. And by Friday, we had the answer. And the answer was he had a rare disease. We had suspected he had, and we found it. It was this thing called IPEX syndrome. And everyone goes, I don't know what that is, but, there's 4,800 of these. So many of us have never heard of these conditions. But they are there, and they're real, and they have a big impact. And what happens in IPEX syndrome is sort of our immune system has to be controlled, right? So we have these cells that come in called regulatory T cells that dampen the immune system and say slow down and act only against things that aren't yourself. And so he had a hyperactive, basically a severe form of autoimmune type of disease. And really the key thing here is that you can only treat IPEX syndrome by transplant. Without transplant, all of the kids with IPEX syndrome have eventually died. So we really needed to pivot, and our transplant team and the immunologists and our genetics team and, everyone rallied around our little hero here and got him ready for a transplant. And I love this photo of Madden. He's post-transplant. He's a bit puffy from the medication we have him on, but he's getting ready for the playoffs, I guess. And has a little Children's Hospital Lego model sitting there on the shelf. And a beautiful institution with nice windows and lighting. So Madden's doing great. And, without that answer, it would have been really difficult to know what to do. And the story gets more complicated or more rewarding in the sense that he actually has a cousin who had the same condition and would have died also without a transplant. So the minute we diagnosed Madden, everyone went, cousin must have the same thing, and he did. And he's also now being prepared for a transplant that will be life-saving.
When you have a question from one of our participants, when you look at a genome, what do you actually see? That those letters that you showed us a few slides ago that would take us our lives to look through, what do those letters represent that you actually see?
Yeah, so there's probably two questions in there, and I'll see if I can decipher which one is being asked. The letters represent in the middle ladder of the code. We always think of the code as that helix that's got those step ladders. The letter is just a code for the chemical base in the middle. And so we have 4 different chemical bases we could put in the DNA. And those chemical bases get interpreted by the body to create an amino acid for each 3 letters. And then we create a string of those amino acids and create a protein. So that can be one interpretation, what do those letters actually mean. What it looks like coming off of the machine might be the other one. What do we actually see in the machine is quite remarkably what they are is just really, really fancy cameras with a sequencing system built in them. So what you do is inside those instruments on a little glass slide, you have millions of small fragments of DNA, and you're chemically adding a new letter code, right? So the next one should be a G. You add a G, and that G fluoresces green. So they all do that. They all fluoresce their own color, and then you take a picture of it. And then you add another, and you take a picture, and you add another, and take a picture. But like you're doing that, right? And so you can do this massively parallel way, so you're sequencing large number of fragments. So that's what we actually see on the instrument, and the instrument interprets those colors as, I think that's a G. We align it to the genome and say, yep, makes sense. There should be a G there, or Every other human we've ever looked at, no G there. They have a C. This patient's got a mutation. Okay, so here we are with Madden. I'm going to switch to another story now, and this is a cancer story. And so Jen was first seen in our clinic when she was, I think, in her late 20s. And she was seen in our cancer genetics program in the clinical genetics unit because she was known to have a family history of hereditary breast ovarian cancer. So this is the BRCA1, BRCA2 stories that people have heard of. And the family was known to have that because there was a family history of breast and ovarian cancer and people had been tested. And her father was known to carry that mutation. So here she is at 29, having to make a decision, do I want to know that? I'm at 50% risk of inheriting either my dad's copy of BRCA1 that's not working or the normal one. If I get the normal one, I'm like everyone else in terms of cancer risk. But if I get the one that carries the cancer risk, I'm sort of at a 60 to 70% lifetime risk of breast cancer and a 20 to 30% risk lifetime of ovarian cancer, and that's quite significant. So the information itself is powerful, and it gives us a sense of that predictive nature of our genome and the personal decisions that go into wanting to know whether we're at risk of this information or not. So Jen decided she did want to know and did test. We call it positive. I don't know why we call that in medicine, because I don't think there's a patient ever told them this news that feels it's positive, but she did have that genetic mutation. So I then get the question is cancer genetic? And my answer is that absolutely, every cancer is genetic. At least every tumor is genetic. In every cancer that's ever been described, there are a very large number of genetic changes that have accumulated in that cancer. Those genetic changes take out the systems that keep that cell in control. They take out the system that tell that cell to die if it's abnormal, to stop dividing. So if you take that system out, the cell's going to divide when it shouldn't be divided. It'll divide when it's ill. It'll accumulate more genetic change. You have a rogue on your hands. So that's cancer. So cancer is definitely genetic in the actual cell. The bigger question I think people are asking me is often, is it hereditary? Am I at risk, or am I predisposed to my cancer? Or is my family at risk of cancer? And so in the diagram I'm showing people is this accumulation of genetic changes that occurs in all tumors, but the normal cells there, which we were born with, did not have a genetic change. But hereditary cancer does happen, right, where people are born with one of those genetic changes in those systems that control cells. Tell them to stop, tell them to repair their DNA, tell them not to divide, tell them to kill themselves. So for individuals who inherit changes in those genes, they have those in all of their cells. So now their cells are kind of, I call them one step. Won't guarantee they're going to develop cancer often, unless it's a really bad one. So for BRCA1/2, you can see that it's one of those genes that's really important for keeping the cell in control. And if you're born with one of those genetic changes, one of your copies is not working, you're predisposed to develop cancer. So we could use the example of breast cancer then and look at how much of breast cancer is genetic. And so we would say that 5 to 10% of breast cancer is hereditary, truly these big BRCA1/2 changes that create a major risk of cancer. There's some clustering of cancer that we don't understand very well. We're starting to find some of those genes. You think back to that early slide around complex traits. Well, there are some complex trait families where there just seems to be a little bit too much cancer. It's not maybe a 60% lifetime risk, but maybe it's doubled, right? That 1 in 10 risk that all women face is now 20%. And what are those genetic factors? Interacting probably with other things that create a cancer risk. And about 5% ovarian cancer risk. So this slide could be repeated for many cancers. Colon cancer looks a little bit like this. And then not for other cancers. If you think of skin cancer is heavily environmental, right? Sun exposure is your big damager. But we do have conditions that put people at risk of cancer. So what do you watch for? And people will say, well, how do I know I'm at risk of cancer? And I say, well, your family history is important there. If you have cancer yourself, the pattern of cancer can be important. Someone who's had multiple cancers at an unusual young age. For breast cancer, we think of bilateral breast cancer under the age of 50, cancer and breast cancer and ovarian cancer. So this is the Angelina Jolie story,
right.
And if you look at our clinic, after Angelina came out with her story of having hereditary breast cancer and undergoing a mastectomy, our cancer referrals doubled. And that was pretty much the story across every cancer genetics clinic. And they were appropriate referrals, which tells us that people out there were at risk. We're just not aware of it, and their physicians weren't aware of it. So that the public messaging, the public health component of genomics, how are we going to do a better job of preventing disease by this type of information? So the story of Jennifer continues in that on a screening MRI at the age of, I think, early 30s, so she would not have been in a breast cancer screening program. She was not known to be at high risk. She was found to have a unilateral breast cancer. She underwent a bilateral mastectomy, in part to deal with the current cancer she had, but also, you know what, that's enough. I'm not ever going to get cancer again. And she went on and had 2 children, we're seeing here, and she's healthy.
Yeah.
And she will not die of cancer, either breast or ovarian cancer, like the female relatives did in her family.
So in this case, we knew the testing helped her to make that decision to have the surgery to prevent the cancer from occurring. Going back to little Madden, we have a question from one of our participants asking, what exactly did sequencing Madden's genome help with? Did it help with better targeting of immunotherapy?
It actually told us that he needed to have a bone marrow transplant. So bone marrow transplant is, we're actually going to kill his own bone marrow system, which the bone marrow populates our immune system, and it does so repetitively. So the stem cells that are sitting in our bone marrow pump out our immune system. So the only treatment for him, we had no immunotherapy, we had no medication that would have helped him. We really needed to get rid of his immune system and give him a new one. And when you give him a new one, so you kill his own one, and then you transplant him, you essentially infuse in bone marrow. And bone marrow cells are amazing. They actually know to go sit in the bone marrow. So they went back and did what they know how to do, is they repopulated an immune system for him. And because the transplanted bone marrow doesn't have that genetic change, he doesn't have the condition anymore. It's present in other cells of his body, but it's not present in the critical cells that populate his immune system. So we've been able to give him a new immune system.
Okay, another question from one of our participants. How do most GPs see genome information? This person's GP, for example, was against it for this person. There's lots of cancer in the family, and the GP suggested that she or he not have the testing. That's another can of worms there.
So the question of how people view this is complex. I mean, medicine is an increasingly very, very complicated field, and you know I can't help but sympathize with frontline physicians who are trying their best and often very dedicated to keep up with a vast amount of change in the healthcare system. So I don't think there's a single answer of how GPs view this. Either some of them may not be aware of up-to-date information, some of them may have personal views, but I think most of them are actually really wonderful. They will call us. You know, being a GP is a little bit like knowing who, you know, you have 3 things you can do. You know, you can call a friend, you can look something up, and so what do you do when you're unsure? And most people know their lifelines. You know, they know who to call and try and sort information out. Sometimes you run into people that have very strong views about genetics. Might be around reproductive risk and testing pregnancies that will maybe try and hold back information. But you know, that's why I think information access is great. You know, people say don't check Dr. Google. I say, well, check the first page of Dr. Google. The first page of web searches is probably pretty good. So if you're unsure, you can find out information. We're trying to do a better job of helping our healthcare professionals develop competencies in genetics across the board. You know, nurses, nurse practitioners, physicians, et cetera, pharmacists, et cetera. Complex question.
And a lot of more information coming almost daily, and I know you've got a lot more information to tell us here.
Sure, so we'll continue on. So I think we're finished the gen story, and the next one is one of my favorite stories in biology, actually. And it's the question of why don't elephants get cancer? And people will look at me and say, well, really? It is absolutely true that elephants, and actually all large mammals, so that can be, you know, whales, rhinoceroses, hippos, anything that's very large has very low rates of cancer. And the paradox, of course, is they have trillions and trillions of more cells than we do. They should have enormously high rates of cancer. Colon cancer should be ubiquitous in an elephant. They have an enormous size of colon, and they live a very long time. So the reality is that humans or mammals have a huge range of cancers. So what I'm showing here is we have patients who have a condition called Li-Fraumeni syndrome. Li-Fraumeni syndrome is caused by having one genetic change in a gene called P53. And P53 is one of those really crucial genes for controlling cells. And it shows you how crucial it is because if you have one copy of it, patients with Li-Fraumeni syndrome are sort of, we say 90% lifetime risk of cancer, but The reality is probably just not all of those patients at some point will have cancer, will have multiple cancers, and will die at a young age from cancer. So the typical human has probably about a 25% risk of cancer, and then we go to the right-hand side of this slide, and we have elephants who are sitting here at their sort of less than 5% risk of cancer. And a remarkable series of papers came out that actually solved this paradox by looking at the genome of the elephant. What is it about the elephant that can protect themselves against cancer? And the answer is that elephants have a very, very large number of p53 genes. They've stacked their genome to protect themselves against cancer. So we have two copies, one we got from our mom, one we got from our dad. Elephants have 40, so presumably 20 from their mom and 20 from their dad. So they double down on their tumor suppressors to allow them to survive this long. And I like this example because it forces us to think a little bit about, well, why do we understand, or even at a place like the University of Calgary or the Cummings School of Medicine, why do we do fundamental discovery-based research? You know, why is it important to look at genomes of other organisms? Or why do we interested in how cells divide and not divide? It's because you never know where your answers are going to come from. I don't know if this is going to be the cure-all to cancer. You know, should we all be doubling down on p53 somehow? Obviously, that's not the simple answer. But there is a cue there, right, in terms of how we might be able to prevent cancer.
We have a question from one of our listeners or participants. You talk about this discovery about the elephant.
Mm-hmm.
The next thing you may think of is, well, how quickly can we get this and test it on humans? And the question is, how far are human trials with CRISPR tech in terms of gene therapy?
Yeah, so I have a slide later on CRISPR. So human trials with CRISPR, so I'll back up a little bit. Our tech-savvy reader is talking about a technology called CRISPR. The CRISPR enzyme occurs naturally in bacteria, actually. And bacteria use that CRISPR enzyme to kill off invaders, for them viruses. So it's their defense system. Some very, very smart people, I think, who are lining themselves up for a Nobel Prize, realize that CRISPR can actually edit the genome. And so you can use CRISPR to go into that specific location. Think of that letter A I was showing everyone in that patient that was abnormal. And you could use a CRISPR enzyme with a guide that would say, go to this exact place in the genome. It would sit right there. And then the enzyme could be used to repair that mistake. And you could repair it the way you want it to. So you can put it back to the same base that should have been there. So CRISPR is being touted as a gene editing or a way of fixing genes. So hugely controversial. I mean, right now there are laws that prevent any research and certainly any clinical trial that would alter the future of genome, right? So we would not ever do this to change a gene that could be passed on to the next generation. We're not there in terms of safety. In mouse models and animal models of genetic diseases, for sure CRISPR is being used to treat things like Duchenne muscular dystrophy, cystic fibrosis, thalassemia. Some of the treatments can be as simple, it sounds easy, you know, pull the cells out of the bone marrow that should be making a little bit of factor VIII to prevent us from having hemophilia. CRISPR those cells, which go in, edit the change, and put them back in the body. And forever, you now, instead of transplanting someone else's bone marrow, you just fix the person's bone marrow, and you have access to therapy. So CRISPR trials will come. There are some very large think tanks in the world, including the ethical use of technology think tanks, science organizations, who are saying, you know, what do we do with these new technologies, and how do we use them safely and appropriately? So thanks for that question.
Here's another interesting topic, epigenomics.
Epigenomics, right.
The environment.
Okay, great. Another big term. So when we talk about mutations, we often think of a change in the genetic code that changes how we produce a protein, right? And those are the genetic mutations. And they're transmissible to the next generation because you've actually altered the genetic code. Epigenetics is the concept that you can silence a gene or activate a gene without changing the letter code and often you methylate it. You put a little cap on it, the way I think of it. When the cap's on, the gene's inactive. You take the cap off, the gene's going to be active. So we now completely understand that epigenetics is a common phenomenon. So I'm going to give you an example, not from humans. If I go into human epigenetics, it's a whole other topic. But these are little Daphnia, multicellular little swimmy things that live in ponds. And when you stress a Daphnia with a predator or no food, guys get scared and they grow a helmet. And that's the Daphnia sitting there on the right. That itself is not that interesting, they just respond to an environmental stress and they change. What is striking is that they pass that on to the next generation. So the next generation of Daphnia also have a little helmet until the stress is removed for a period of time. And then they can reverse it. And so what's happening here is epigenetics. You're not changing the genetic code, but you're changing the expression of the genetic code based on an environmental stimulus. So in humans, the concept of epigenetics is really exciting right now because it brings together the concept of gene-environment interactions. And people ask, nature versus nurture, and I say, well, you know what, it's probably both. And sometimes it's more heavily nature, and sometimes it's more heavily nurture. But this is the link maybe between the two. So can environmental experiences, can life experiences alter our metabolism? Can it alter the next generation maybe? One of the clues we have in humans, just as an example, was the observation that babies who are born small have a much higher rate of adult onset chronic diseases, particularly hypertension, diabetes, heart disease, that triad of metabolic syndrome and obesity. And what's probably happening is the thing that's adaptive to adjusting how the genome works and how our physiology works as a small baby, because the placenta is not working or mom is severely malnourished, makes the baby adapt to that environment and be small. That's adaptive as a baby. Helps survival for sure. But it's maladaptive because now you've reset those thermostats in an epigenetic manner. So epigenetics, we're going to hear a lot more about it. And there are ways of sequencing for epigenome so we can analyze the epigenome. The tricky part is that each one of our cells and each one of our organs has a separate epigenome, right? It's very dynamic. And it changes over time. So epigenetics will be a complex area, but really exciting.
Yeah, we're going to learn a lot more in the coming years on that one, aren't we?
I think so. So we're also heading into an era of designer therapies. So these are medications that are being created to target very, very specific pathways. So we have cancer drugs that are targeted to very specific molecules that are abnormal. But the one that's going to hit a lot of us, I think, is this concept. And this one is known to all of us, just about all of us, that when we're prescribed a medication, there's going to be one of four outcomes. And ideally, you're going to be at the bottom right-hand corner of this, which means the medication is not going to create side effects, and it's going to make us feel better. And, these are the medications that people think their doctor is brilliant. They're on it, they don't know they're on it, and their symptoms went away. The people that are the most upset are at the right upper corner. Their drug is giving them side effects, and they get no benefit. And they're not happy at all. They're going to stop that medication. They're going to feel like they wasted their money. And then there's every other combination. So you can be on a medication, get some benefits, but there's side effects. Do I want to tolerate the side effects? And then maybe I'm getting side effects, but it's not helping me. Or I get nothing at all, sorry. So not toxic and non-beneficial, I'm on the medication. So this is pharmacogenomics, the concept that part of the answer to this perplexing problem of why we respond to some medication and other people don't respond and get side effects, et cetera, is that our genes are different. How we metabolize our medication is different. How sensitive we are to medication is different. And so right now it's estimated that maybe 20 to 30% of all prescriptions written would be improved. The outcomes would be improved if we had access to good pharmacogenomic testing. And that would mean that we would decrease drug costs, we would improve outcomes, satisfaction, et cetera, et cetera. And then the simple example I use sometimes is a common painkiller called codeine. So codeine is part of Tylenol 3. We actually know that codeine is not the active part. Our body takes codeine and metabolizes it by an enzyme system into something that's more like a morphine-type derivative. And that's the one that gives us the pain control. Well, 10% of us don't actually have a good enzyme system to do that. So 10% of us get a Tylenol 3 post-operative pain control, and it does nothing for us, right? It also doesn't make us constipated because it actually does nothing. Our body doesn't activate it. So the side effect rate is low, but the beneficial rate is terrible. 1 in 1,000 to 1 in 5,000 people have an enzyme system that is ultra rapid. It's super fast at metabolizing that codeine to the active form. So that person, think of a young child, post-op tonsillectomy, a little bit drowsy, a little bit swollen throat, had a long day, is on a Tylenol 3 for pain control, and is a hypermetabolizer and gets toxic on the medication. And there have been kids who have died from a single dose of Tylenol 3. In fact, there's an example of a mother who was on Tylenol 3 breastfeeding a baby who was transmitting that medication to a baby who was a hypermetabolizer who died. So codeine is no longer used in children. There's recommendations to just stay away from it. And there are lots of other more important medications that you know, antidepressants, et cetera, where the understanding of pharmacogenomics. So do I think we're ready on this one? I think we're getting there. I think we're going to start seeing pilots. We just recruited a world expert on pharmacogenomics for the Cummings School of Medicine who works on mental health drugs to help us think about how we're going to further our understanding of the role of pharmacogenomics and how we might move this into clinical practice.
Is this, would you say, 10, 20, 30 years away?
I think it's shorter than that. There are pilot studies out right now in a number of clinics. I think the model is not clear. You know, do we do this at the pharmacist level? Do we do it at the prescribing physician level? The role of electronic medical records is really important here too. You know, where does that information reside? So that all physicians, you know, we change physicians all over the place, and part of it is improving communication. So where does that information reside?
Who has access to it? Is it the kind of thing that an insurance company could have access to, or an employer?
Well, we need to think about protecting privacy of genetic information for that reason itself, right? So, you know, pharmacogenomic one might not do a lot of harm to us, but there are other genes that could, in terms of people wanting to say, well, maybe I don't want to hire you, or you're at high risk and I don't want to insure you. We do have to think about protecting privacy for sure. So this brings me to sort of the third topic in our series, which was, you know, where is medicine heading? And we can think of medicine having had a few phases. In the first phase of medicine, I think our dean, John Meddings, describes as intuitive medicine. You know, that was the good family doc who had a lot of experience and just seemed to know that this was probably going to be better than that. You could go see another doc and they might have a different intuition, and who was right they would fight about, but there wasn't good evidence. But then in the sort of 60s and 70s, and really coming to light in the 1980s and onward, and the era we're currently in is evidence-based medicine. So this is the era of the randomized controlled trial. So I take, you know, 500 patients with diabetes, and I treat some this way and some that way, and I see what the outcome is. And if group A is better than group B, then group A, the treatment that group A got is the better one. And we did this for cancer treatment. We did it to prove that people should be on anti-cholesterol medication, that people should be on antihypertensive, that this epilepsy drug is better than that one. And so this was evidence-based medicine. But you look at my picture here, you know, it's a bit of a fuzzy era still. You know, we're not as good as we would like to be. And I think we have to admit that our outcomes aren't always as targeted as we would like. And part of the answer is that, well, we're all unique. We are tremendously unique. I would say that absolutely everyone for sure has a unique genome. And what influence it has on sort of our risk of disease and our response to disease. I've already told you we probably have all unique epigenomes, and we all have unique environmental exposures, and we have unique health histories. So we talk about the concept of precision health or precision medicine as being, let's accept that we're all different, and then how do we start understanding our differences? And so adding in information, the more accurate information on our genomic risks or our genome drug response, et cetera, our risks of disease, can give us tailored therapy. And I think Jen's an example of that in many ways. You know, we targeted her screening in a very specific way because of her individual risk, and we got a good outcome on it. The population health trials, the randomized controlled trial, would have told us that Jen, at the age of 31, should not actually get breast cancer screening. There's no benefit for it in 31-year-olds. But that's not her. That's 1,000 women who are 31. So how do we individualize risk? And then, more importantly, how do we drive this to better therapy?
We have a question from one of our participants. It's a little technical, sort of going back to what you were talking about a second ago. At least 40% of pharmaceuticals act through G protein-coupled receptors, and those receptors are expressed on primary cilia. Do you think that part of the pharmacogenomic issue is a primary cilioma issue, which I guess you're going to have to tell us what the question is before you can answer it?
Yeah, so whoever's asking is a very technical person, and you should probably take it offline. Reach out to me if you can find me, and I'd be happy to go into more details. But the question here is basically the cells are incredible machines, and there's a structure that we have in our cells called cilia. And outside of cilia, these are the little beating things that are sitting in our lungs and push mucus around and make sperm move. And so we have little motile cilia, but we have immotile cilia that are really sensors. And the activation of those sensors by environmental cues gets signaled through a system, and there are genes in there, the G-coupled genes. And is that the mechanism is basically what I'm being asked? And I don't think we know. There have been some genes identified, I don't know off the top of my head, that are responsible for the pharmacogenomic responses. Often they're more in the CYP pathways, the CYP pathways are the genes we use to break things down. So they're detoxifying type of enzymes. And obviously our body just uses them. They didn't develop because we didn't have these medications when those enzymes were developed. But now our body's using those same systems to detoxify ourselves or to break down medication or to activate them. So
the technical, yeah.
Very technical.
Well, we have another question that's a little more general. Do you think that governments will be open to covering the cost of tailoring medicine?
Yeah, so that's also an important question. And, you know, I make this sound easy, right? So change in healthcare is difficult, and in some ways it should be difficult. We have to be able to prove that what we're changing to is better. And these days, in the context of being cost-constrained in the healthcare system, I think we have to only add new things when we remove other things or when there's a significant benefit. So I think governments are interested in this. We have a precision health initiative in the province that's bringing together our two academic institutions, Alberta Health Services and Alberta Health, to think about how we're going to prepare and start evaluating and implementing, if appropriate, the precision health concepts. Some of it is going to be slow, and some of it I think is going to be pretty ready to go.
And then there's the whole ethical side of things and how much information, who gets access to that information, how to protect that information. That's a pretty big concern, isn't it?
Yeah, it is a big one, and I'll come to sort of a Genetic Privacy Act slide in a second. So I guess what we've been talking about really is a disruptive technology. And so there are think tanks in the world that brood about, you know, what is going to have an impact on health, on economies, on society. What are these disruptive things? You know, the nuclear power type things, the telephones, and so the current ones are things we've all heard of, you know, driverless cars, the Internet of Things. But one of those is next generation sequencing, the ability to sequence genomes, and not just human genomes, you know, genomes of our agricultural species, of our agricultural animals. But in particular, I think the human genomes and the models of human diseases that we use are already having a huge impact. And we're just at the beginning days of it, of trying to understand this technology. And I think coming to your question, Jennifer, a little bit is, you know, why do we need a law to prevent genetic discrimination? And Canada is one of the only G8 countries that does not have a genetic discrimination law or a law that protects the uniqueness of our DNA, which is really unique to us. And so one is in front of Parliament and passed, and we're going to go to Senate and pass, but it's so controversial for a number of reasons in Canada, and we really hope it does pass. I think the ability for people to undergo genetic testing, understanding that I'm learning something that's unique about myself, and I want to harness the power of that to help my physician understand how to treat myself. And I think even individuals want access to this information. So we do need genetic privacy protection. I have a slide here, and we talked about CRISPR a little bit earlier, and to me, CRISPR is just one tool to change the genome, and we have other tools, gene therapy. And in Cummings School of Medicine, one of my colleagues, Dr. Khan, just participated in a clinical trial for a rare disease called Fabry, where we pulled that patient's cells out of his body by selecting them, and once we had them out of his body, we fixed them. We used a virus that could re-inject a new gene that could produce that enzyme that he was missing. And we've put them back into his body. And so there are other ways of fixing those genes. We can edit them. We might be able to go in and use gene therapy, or we might use targeted medication, right, for a very specific defect in that pathway. And again, those are huge technologies. How do we use them safely? But they're potentially life-altering. So now I can't resist getting a bit of feedback from the audience again. So we're going to go straight to a poll. This was another poll in the Globe and Mail series. And the question here, which was a little bit more complex, and I'm just going to simplify it a little bit, is, gambling with our DNA, would you share your genetic profile? So if you're kind of for the concept of, yeah, I would have my genome sequenced, then I would make my health information available to make it better at interpreting that, because we need lots of information here on what the individual variations are and what contributes to disease or not. So if you're thinking, yeah, this sounds okay to me, and maybe I'll put the caveat with the genetic privacy law in place, but am I kind of okay with this stuff, or are you kidding? No way would be your no. So let's see what the poll feels like.
Yes, this will be very interesting because the ramifications of that information, your genomic information being out there, as we mentioned before, to employers, to insurance companies, to hackers, who knows, versus the possibility of really advancing your precision, your healthcare, your own personal healthcare.
We're just watching the numbers come in, and here we can have a look. So interesting. So about
it's not a slam dunk, is it?
7%, 32%. Yeah, it's interesting depending on what perspective you take, right? You could say, well, that's telling me that 2/3 of the people online here, and if they're representative of the Alberta population, that's a big task. You have to figure out how to get this information to 2/3 of people that are really seeking it. But also that there's maybe appropriately a healthy group of people saying, no, wait a minute, certainly before doing it, I want to know what I'm getting into. So when we use genomic testing right now clinically, we for sure spend a lot of time with these individuals, helping them understand the advantages and potential disadvantages. And in our research studies, they participate because there's a protection under the sort of research ability to hold on to information. But once it gets into the healthcare system, it's not as protected. So interesting answers. We're going to show you actually how the Globe and Mail readers responded. So now again, they didn't quite get a yes/no question. What they could do is choose somewhere in the grid, and if you're choosing up towards the top, you're more likely to be interested in having your genome sequenced and share your information. Those two questions on the kind of up and right-hand side, and if you're kind of more negative or a bit more worried about this, you're more no. And you can see the Globe and Mail readers were also somewhat at the top. And maybe it's a little bit more awareness through talks like this that make people not only more enthusiastic, but also maybe in some ways a little bit more wary. And I'm okay with that. We're not here to sell anything. We're here to understand it. And so I do look at genomics a little bit this way, which is that it has a number of features to it. It is a powerful technology. We can think of it as being a component of the future of healthcare, and as costs continue to drop, I think access will become more ensconced in healthcare delivery, and for sure it's a common research tool. It is predictive, it's more precise, so it will drive this precision medicine era. It's disruptive in the sense that it makes us think about the power of the technology and how to use it appropriately. And it's highly personal. And when you're dealing with something that's highly personal, you worry about questions of discrimination and the privacy of that information. So I view this as many technologies that humans have developed. There's potential to it and great opportunity, but we have to think of how to harness that. So 10 years ago, I guess 5, 6 years ago, when the Human Genome Project hit its 10-year anniversary, there was this concept that, the future was going to be bright for genomics. And I have to admit, there are things that we're able to do now in terms of diagnosing patients and arriving at specific therapy that I would have only dreamed of in my career. The ability to actually sequence through and arrive at answers for people who struggled for their whole life to try and get an answer is a very powerful thing, to see them go on to a very specific therapy. So we're in a fascinating era.
Is it available to the average person at this point? Can I walk into a clinic somewhere and put down my money? And
I would say no. So there are commercial labs in the U.S. that will offer this on a fee-for-service type of basis. Physicians still need to order it. And I would say for the walking well, we get this question a lot. It's under study right now. For the walking well, getting a whole genome sequence is probably not going to be beneficial. It will come, and I think as we get better at interpreting the genome and getting predictive information out of it, if people have a specific health issue and are wondering if it's genetic, yes. We're struggling with keeping up with the demand, and the complexity of the clinical care that we have to provide has gone through the roof. But people should reach out if they think they have a medical issue that could be genetic or would benefit from testing, and the healthcare system will do their best to help them out.
Someone's asking, certainly you see the ads for the direct-to-consumer healthcare companies that are offering some genetic testing. What are you worried about the type of information people are getting from these sort of companies that they may or may not understand?
I do a little bit. I view the information as sort of, there are things that are fun to know, ancestry and, where my family might be from, or some of the traits I have, maybe they're unique in my genome, maybe I can see them in my mirror, but, so there's things that are fun to know. The things you might want to know, there might be some predictive information in there that could be useful. I think what scares me is the things that maybe people don't want to know or aren't ready to know, right? So buried in that same task can be all of those. So am I ready to find out I have a BRCA1 mutation? And if you are, great, then maybe you should access it through the healthcare system if there's a good reason. It's not a perfect screen. And if you're not ready, are you sure that you want that information and that it's going to be protected? So it's a little bit of buyer beware in terms of what you're getting. There are lots of people predicting, I would say, all kinds of things that are sounding a little bit too good. I can find a great partner by, genome matching. Okay, I'm not so sure about that.
That one smells a bit like snake oil, doesn't it?
Yeah, athletic performance based on genetic testing, not so sure. Nutrigenomics, a little bit unsure. So, there's people are going to try and use this information and sell it. Okay, so I'm going to wrap up a little bit. So we've
got a lot of questions that we didn't get to, but we do have this. There we go. I think you and I are both hitting the slide at the same time.
Okay, here we go. So key messages. To wrap, we talked a little bit about the 3 billion letters of our genome, and that it contains all the instructions necessary for development and maintenance of our body. And therefore, it wouldn't surprise us that genetic changes would impact us, both in terms of development, so birth defects and disorders of childhood, but also how our body functions. Genetic variation has a large impact on our risk of disease, but also in our response to treatments. We talked a little bit about this pharmacogenomics and targeted drug therapy. The rapid advances in sequencing, but also the computer power behind all of this is very powerful, and that human genomics will be one of the cornerstones of this new precision medicine era. So I put some resources here. If people want to have a look, you can look up the NHGRI Genomic Medicine and Healthcare as a web search, a brief guide to genomics, our Children's Hospital Research Institute, and the university department. Google away with any of the terms in the Wordle will probably get you to the right place.
That is a lot of information. I'm not sure if that slide made it up with the links, did it?
Yep.
Okay. Wow. There's so much to think about with this topic. Thank you very much for coming in and starting us to think about it anyway. There were many, many questions that we didn't have time to get to. There was so much information that we were putting out there. So thank you, everybody, for the questions. And we have more webinars coming up in the next few weeks. Our next one is nanomedicine, the new way to detect and treat cancer. Then we have another one about clinical depression, what you need to know, and microbiome, why a few trillion germs can be a very good thing. So check out ucalgary.ca/explore to learn more about those webinars and other information that we have up there. And I want to thank you all again for joining us today and for your excellent questions. And thank you again, Dr. Bernier. And my name is Jennifer Alford, and we will see you next time.